Welcome back from lunch, everybody. So we're kicking off our second part of the exposure section of this with Marc Verhougstraete, who's also from the College of Public Health here and speaking about combined methods for relating human health with contaminant mixtures in water. So, thanks, Marc.
Nice. Thanks, Frank. So I'm going to jump right in. Traditionally, the monitoring, managing, and mitigation of water contaminants has been performed one chemical or one contaminant, one state of contaminants, relating back to a single health outcome. But we know that this is completely inaccurate. We are exposed to complex mixtures in our drinking water and our surface water every time we enter that environment. And so within those multiple contaminants, it requires multiple experts to work together on the exposure side. And those multidisciplinary teams are necessary to understand the human health components of these complex mixtures, as well as the animal and environmental incorporations of them. The last One Health Symposium, Linda Birbaum was here, and she mentioned that pathogens make up the environment and are part of the exposure. But she also indicated that NIEHS does not fund pathogen exposure research because that's NIAID's job. But NIAID focuses on infectious diseases from a clinical and treatment standpoint, and they don't fund chemical mixtures research. So we're kind of stuck in the middle here of where do we find true chemical and microbiological exposure funding? And so up to this point, those complex mixtures have been done independently, and we continue to move forward on that route where we are assessing microorganisms and metals from a independent standpoint. But now this interdisciplinary teams that we're developing here start to get to this point of trying to incorporate complex organic, inorganic, and biological contaminants in water. So this framework that we use to understand these complex mixtures is called the exposome. It requires multi-disciplines to come together, but this still only focuses largely on human health. And we're here at a One Health conference. We need to start thinking about the exposome from a One Health standpoint, where it's the complex of the environment, complex of the animal health, and human health all integrating together. So the goal for this talk today is to demonstrate how the exposures to contaminant mixtures from animals and the environment impact human health outcomes. So we're going to talk about how the U of A is developing these multidisciplinary teams to address contaminant mixtures and relate them to human health outcomes in 3 geographical areas. The teams that we've developed here are ecotoxicology, microbiology, and epidemiology. The ecotoxicology group is being led by Frank von Hippel, whom we all know. Frank has merged together ecotoxicology with toxicology mechanisms and health disparities by integrating wildlife animals with lab animals, stickleback fish that we heard about earlier, to really assess health outcomes at a local level. The health-related water microbiology side, I'm leading that team, where we use targeted and non-targeted culture and molecular methods to assess the microbial water quality, then integrate that data into risk assessment that does combine metals and microorganism dose response models to really understand these cumulative health risks. On the epidemiology side, we have that team being led by Dr. Robin Harris and Dr. Heidi Brown. Heidi Brown is a spatial epidemiologist who looks at the transport of vectors and infectious diseases throughout the environment. And we have Dr. Robin Harris, who is a community-based epidemiologist who looks at the prevalence and the occurrence and prevention of cancers. Together, our teams are coming together to assess the cumulative health-related environmental exposures, and in this case, we're trying to relate that back to water. So I'm going to talk about 3 case studies that our teams are coming together to address. And the first one is Lake Etienne. This is Guatemala's largest lake. It's about 25 square kilometers. And within this watershed, there's 180,000 people who rely on this lake for drinking water, for food, for irrigation, for sanitation, and for transportation. Over the last few decades, there's been an increase in agriculture, chemicals and nutrients, as well as untreated sewage, leading to a lot of nutrients in this lake, causing eutrophication. Dr. Frank has been working down in Guatemala for a number of years, and his team, along with the group that's already down there, has looked at physical, biological, chemical aspects of this lake. They also looked at the occurrence of pesticides in the community and how they're manifesting into human health and to the water. And they're finding specific insecticides and fungicides that are really high concentrations in the lake, and these are likely biomagnifying and bioaccumulating in wildlife and humans. So there's a lot, though, that we don't know, including the extent of environmental contamination from a microbial and a metal standpoint, and how these complex mixtures manifest themselves into human health. And so last summer, we had a group of students down there collecting some environmental samples who are from the University of Arizona. And the next steps will be this next trip that we take down in the fall, where we hope to start to measure food web dynamics of toxic metals, including mercury and pesticides, into snails, crabs, and fish within this lake. We hope to then take a biological assessment where we start to look at pathogens and cyanotoxins in the surface water, in the drinking water, and the used water. We hope then to also do a source apportionment so we can start to prioritize where efforts need to be put in place throughout the environment and throughout this watershed to have the greatest effect on reducing these contaminants in the water and in the humans. We also will deploy some health and behavioral surveys to really understand what other factors may be driving health outcomes in this group. That data will all be honed together to develop a risk assessment on these complex mixtures. And so we're taking our first trip down there this fall, or I'm taking my first trip down there this fall, and hopefully we can start to move this project forward with a PhD student who's joining us at the U of A starting this fall from Guatemala. So she's got a personal interest in this project, and we will be able to move that forward really easily. The second group that we want to focus on in the second community is Tamaulipas, Mexico. This is a group that formed when about 3,000 immigrants were unable to enter the United States. And so they set up their community on the northeast area of Mexico, along the Gulf of Mexico. This group has faced a lot of challenges, as you can imagine. One of them is environmental contamination. They're subsistence farmers, and so as such, they're harvesting a lot of the fish in these estuarine systems. And one of the issues with this group, though, is that there's a lot of industrial contamination found in the fish. The University of Tamaulipas has found cadmium, lead, and other heavy metals in the water, in the soil, and in the fish of these in these communities. And so there's already health concern associated with subsistence farming and harvesting in these communities. But what we truly don't know is the contaminant mixtures in fish, water, and soil. Beyond that, there has been virtually no animal, environment, or human assessment for pathogens in this community as well. So while we're down there, we will be looking for health associations using surveys again. We'll be assessing chemicals in food, water, and animals. We look to characterize the perceived and actual health outcomes through these surveys, and then quantify the health risk from identified organisms and exposure pathways, again with a new PhD student who's going to be focused on this group starting this fall. And the third project that we want to talk about is focused on southern Arizona in terms of gastric cancers. We know that gastric cancers disproportionately affect Hispanics throughout the world, and the leading cause of gastric cancers is H. pylori. H. pylori is transmitted via the fecal-oral route from contaminated food and water. And we know that in the presence of metals such as arsenic, H. pylori causes increased infection and cancer stem cell markers in gastric systems. What we don't know about southern Arizona, though, is the H. pylori infection prevalence. We don't know the quality of drinking water in our unregulated or rural wells. And we don't know what social or behavioral determinants of health may be driving gastric cancers in these communities as well. So this project's being led this summer by Taylor, our master's student over here. She's driving this really awesome group this summer where we're going to be assessing H. pylori infection status throughout southern Arizona, We'll be quantifying metal and microorganisms in drinking water throughout the southern Arizona communities. And then again, we'll be surveying health and behavior that may be driving the gastric cancer risk. So in wrapping up, the exposome focuses on human health, and it requires many fields in order to really identify an individual's health outcome associated with the lifetime exposure of contaminants. But we don't have the computational power, we don't have the human knowledge to actually fully understand the exposome at this point in time. But we have to start somewhere, and because of that, our team, our multidisciplinary team, is out collecting biological information and metal data, metal contaminant data, in our animals, in our food, in the water, our environment, and in the humans. And so because of this, we're hoping to build this framework, this approach, this data that can be used into the exposome models in the future, as well as bolster up our One Health pillars so that the animals, environment, and humans all can be more resilient in the presence of these mixture contaminants. And we're hoping that the data that we are generating can inform environmental health and policy that is very targeted to unique communities and based off of the true mixtures that they're being exposed to and the health outcomes that are associated with these complex mixtures. Oh man, Frank, I updated the slide for you and I included your information, but it didn't make it. Sorry. Frank is also co-presenting on this. So.
Great. Thank you, Mark.
All right.
Do we have time for some questions for Mark?
Yeah, John.
Mark, do you know of any of these chemicals that would enhance the infectivity or the seriousness of any illness caused by a microbe? You know, there was speculation 20 years ago that some pesticides in combination with viruses caused more serious infection in people. Is there any evidence for that today?
Yeah, that's kind of the cutting edge of what I'm working on right now, as well as the H. pylori. We're using stomach organoids, so gastric organoids, which are biopsies from human stomachs. We're growing those up as 3D models and exposing them to various concentrations of arsenic and then infecting them with H. pylori. And that's how we know that with increasing concentrations of arsenic, we're seeing increased opportunities for the H. pylori to cause infection, to attach to the epithelial cells of the stomach, and to actually cause cancer. So that's exactly where we're heading as well, because we can all do the chemical mixtures or we can do biological mixtures, but moving those two together, we know there's multiplicative effects that are happening. And that's kind of what Frank and I are aiming to do. Yeah, good question. Question in the back.
Yeah, my question is, how safe is it to do research in Tamaulipas? I mean,
yeah, that is a very fair
scary.
question. I actually brought it up to Frank here a couple of weeks ago. The area that we are going is safe. The route that we're taking is considered safe. We'll be flying, we'll be traveling with the government and with the universities down there. And so they've reassured us that it is very safe for us to be there. Yeah, great question.
In your southern Arizona H. pylori study, do you feel an obligation to treat the individuals with the triple antibiotic regimen usually used to treat H. pylori?
So we have a NIH R01 that we're preparing for submission here later this month, and that is the conversation we had this morning. We will be disseminating the results from the water quality and the H. pylori test. We're using urea breath test, which is a portable test that we can give H. pylori infection status to, not quite real time, but within a couple hours. And so I can't speak to the clinical treatment of it, but we will be providing for anyone that tests positive for H. pylori treatment options, as well as the regimen that they can expect from that. Yeah.
Okay, thank you, Mark.
Yeah, you're welcome.
I love how the questions are all exactly where he is with his research project. So thanks very much. So we'll get our next speaker.